Okay, can everybody hear me okay? Cool. Okay, so I'm going to get cracking a little early because I have 15 slides for 15 minutes, which I think is a mistake. Okay, so my name is Mariano. I'm a postdoc in Kathleen Donahue's lab. Thank you guys for sticking it out so close to the lunch break. Today I'm going to give kind of a high-level overview of some of my postdoc work in the Donahue lab, but I'm more than happy to talk about methods or the nitty-gritty offline after. Okay, so of course all great talks start with a big unanswerable question, so this one's mine. Where does variation come from? I think we're all interested in that. So of course we know that genetic variation and changes in allele frequencies create trait variation that we're interested in. But variation also comes from the environment, and that's expressed in our sort of classic equation where variation in trait, which is y here, is equal to genetic variation and environmental variation and the interaction between the two of those. And it's really that G and that G by E that I'm interested in studying. Because that's the part that represents phenotypic plasticity. So I'm just going to take this opportunity to define phenotypic plasticity, and that's the ability of a single genotype to alter its phenotype in response to an environment. If you have any other definitions, I'll give you a second to just forget them now. Okay, thank you. This is the correct definition. I won't be taking questions on it. Okay. So when might phenotypic plasticity be adapted? So let's imagine a pretend population in a pretend environment, and let's say that its environment changes. Let's say it changes in its temperature, so the environment goes from cold to warm. This is kind of the situation where you'd expect to see local adaptation. Favorable alleles that code for heat tolerance will be selected for, and then the population will be able to tolerate heat, and then they'll be happy. But what happens if that environment then changes? What if it returns back to a cold environment? Or what if it fluctuates between warm and cold environments? This is kind of our classic setup where phenotypic plasticity might be adaptive, because in this case you might imagine that organisms need to have alleles that allow them to tolerate both the warm and the cold environments. But one interesting thing is this x-axis here that I didn't get to talk about yet, which is time. So of course, if the environment is fluctuating within the lifespan of an organism, they would of course they would need to be able to tolerate both the warm and the cold environments. But let's imagine that this time scale is just a little bit longer. And let's imagine that the environment is actually fluctuating just beyond the life cycle of an organism. So let's imagine that this organism grows, or perhaps germinates, just tossing out ideas, in a cold climate, and then it's an adult in a warm climate. And then the next generation germinates in a cold environment and grows as an adult in a warm environment. Here, the periodicity of the environmental cycle extends just beyond the lifespan of the organism. And this creates a really interesting scenario because a single organism will never experience both the warm and the cold in its lifespan. And because it germinates in the cold, its cold environment might not actually be a good predictor of its adult environment up here. And in fact, in this scenario, the previous generation's adult environment might actually be more predictive of the offspring's adult environment than the early life conditions. So in this case, we get an interesting type of plasticity called transgenerational plasticity, where it might be actually advantageous to respond to the parental environment rather than your own early life conditions. Okay, well you might be saying, Mariano, that's a little contrived. When would we expect to see that condition? So I'd actually argue that we see it all the time in the form of seasonality. So organisms that live over short periods of time, perhaps an annual plant, might actually experience this cycle that extends just beyond its life cycle. So for investigating this question, we use our favorite model organism, Arabidopsis. You may have heard that Arabidopsis does not have an ecology. Those are vicious lies spread by our enemies. Actually, Arabidopsis has a great ecology. It's a winter annual life cycle. And really importantly for our research, its life history transitions, the transition to reproduction, which is bolting, and then flowering, and then the development of fruit, actually depend really heavily on seasonal cues like temperature. Just so happens. And we already know actually that thermal cues alter phenotypes, both within generations in the form of plasticity, and between generations in the form of parental effects. So Arabidopsis is actually a really great system for looking at this. Okay, so what were our goals for this big project that I'm working on? We really want to identify variation in the modes and components of transgenerational plasticity, which I'll talk about in a second. We wanted to identify genotypic variation in the expression of transgenerational plasticity. And we also want to understand the molecular basis of transgenerational plasticity. We'll talk about those in turn. And to do that, we developed this really complicated experimental design, which I will definitely walk you through. You don't have to try to understand this on your own. But what's happening here is that in the first generation, we took a bunch of genotypes, 8 genotypes actually, and we grew them in either warm conditions or cold conditions. Then we took the offspring of those plants and put them in either warm conditions or cold conditions from the warm parents, and then from the cold parents as well in warm conditions or cold conditions. We also did a common garden experiment, which I'll reference just a little bit, but I won't really dwell on it so you don't have to think too much about it. So anyway, by the time we get to the third generation, we have environmental sequences where any given group has experienced warm, warm, warm conditions, or perhaps cold, warm, warm conditions, and so on factorially. And this design is actually really nice because we can test the relative influence of within generation signals, those offspring signals perhaps from early life, versus parent signals versus grandparent signals. And that sets up some interesting predictions because it seems intuitive in the case of cycling seasonality that you would want to respond to your parent conditions. But maybe it's not so good to respond to your grandparental conditions because that was kind of far away. Maybe your grandparental conditions aren't actually predicted because they were farther back in time. You also might imagine that your offspring conditions also have some predictive value. So you might not expect, even if you see the expression of transgenerational plasticity, it might not be stable in all possible offspring environmental conditions. So we wanted to test that as well. Of course, this involved growing an insane number of plants, and this would not be possible without our heroic lab tech, so I just want to acknowledge him here. Okay, so as I mentioned, there are these components of transgenerational plasticity that are kind of set up by our predictions of what we might see. So we kind of thought about how transgenerational plasticity might manifest. So in this first scenario, we exposed the first generation to two different environments. And regardless of the next generation's environment, we still see a difference between the phenotypes based on the parent environment. We might think of that as transgenerational plasticity that's able to be induced. But whether it's persistent or not actually depends on whether it lasts until the third generation. So we wanted to assess one more generation, and then if we see the induction of transgenerational plasticity that lasts, it's both inducible and persistent. And then if it also survives, even when we expose the offspring to new environmental conditions, we might also think about it as being environmentally stable as well. So we tested each of these components independently because we could with them. Okay, so we measured a lot of different phenotypes. We looked at rosette biomass phenotypes, which is leaf member folding and rosette size. We looked at phenological measurements, as I talked about, which is time to bolting, time to flowering, time to fruit maturation. We looked at germination phenotypes, which is germination propensity. And of course, we also looked at molecular phenotypes because we really want to understand the molecular basis of how transgenerational plasticity might be manifest. So we did gene expression, DNA methylation, and we looked at small RNAs. I'm only going to talk about the DNA methylation today because actually gene expression data is pseudo-aligning on my laptop at the Airbnb. Okay. Okay, so what we end up with is figures that look like this. So on the top, I'm only going to show you two genotypes here. On this top panel here is Columbia, on this bottom one is Landsberg. And here are the environmental sequences that they've experienced across generations. So by comparing these box plots, for example, if we compare this one over here with this one right here, we can see the grandparental effect. And in this case, the grandparental effect isn't very big. If we compare this one with this one, we can see the effect of the offspring environment, which in this case is actually quite big. And then if we compare this one with this one, we can actually see the effect of parent environment, which is also actually quite large in this case and in the opposite direction. And we can construct this whole thing as an ANOVA, and what we find in the case of, so this is germination propensity, germination propensity on the y-axis. And in this case, we actually found that parent environments influence germination propensity much more even than offspring signals. So this is strongly inducible transgenerational plasticity. One interesting thing is that we actually didn't find it consistently across all genotypes, which suggests that there's genotypic variation for the ability to express transgenerational plasticity. Okay, we also saw differences in the stability and the persistence of transgenerational plasticity. So here, just to orient you, on the x-axis, we have time to each phenological event, so bolting, flowering, and first mature fruit. On the x-axis is the days since planting. So differences between these two lines are differences due to the grandparental environment. So here we're looking explicitly at grandparental effects. And over here, these different panels represent different environmental sequences. So in this case, generation 2 was cold and generation 3 was cold. And here, generation 2 is cold, but generation 3 was warm. And we can see that in some cases, like here in Landsberg, we do see that there are differences in time to bolting and time to flowering as a result of the grandparent environment. So this is actually a persistent transgenerational plasticity. But you'll notice that it doesn't actually appear in all environmental sequences, which suggests that it's actually quite unstable, which is actually what we would predict. We would predict that in some cases you would want to respond to your offspring environment. So here, these little asterisks are significant differences, and we do have significant differences, but not in all environmental conditions and not in all genotypes. So there's genotypic variation for the induction of transgenerational plasticity. There's genotypic variation for the stability of transgenerational plasticity. And there's genotypic variation for how long it lasts, which is the persistence. We also looked at DNA methylation, and I'll just kind of breeze through this, but I'm happy to talk about how we did this offline. But when we look at the influence of parent environments on DNA methylation, we also see a lot of inducible transgenerational plasticity, which means that DNA methylation patterns were different in seeds as a result of their parent environments. Interestingly, and just as a side point, you might imagine that it's a case of quote-unquote epigenetic inheritance. We actually looked for that, and in this case, we didn't see a lot of evidence that that was the case. We saw new induced patterns that weren't necessarily symmetrical with the parents, which suggests that they're not actually being inherited with fidelity, and we actually suspect that this represents the involvement of a broader regulatory pathway, perhaps RDDM, but again, I'm happy to talk about that at another point. Okay, so just a quick summary of what we have here. Transgenerational effects are pretty unpredictable, and that's because they really depend heavily on the present environment as well as the sequence of ancestral environments. Usually, they tend to decay over time, so mostly we found that grandparental effects were weaker than parental effects, which suggests that decay over time. But in some cases, we actually saw an acceleration of transgenerational plasticity over time, where grandparental effects were actually stronger than parent effects, depending on the environmental sequence, which was actually quite surprising to us. And so, as I mentioned before, genetic variation exists for the induction, the persistence, and the stability of transgenerational effects, which sets up kind of a G by E scenario. I'm really sorry to introduce that concept, but here we are. And our data suggests that each of these components, because there's genotypic variation, that has the potential to respond to selection and evolve. We also tested, I didn't show this data, but we tested the correlation between the effect sizes of each of those components of plasticity, and they're actually not very strongly correlated with each other. So the degree, how strongly transgenerational plasticity is expressed, doesn't seem to be correlated with how long it persists. So that suggests that each of these components might actually be evolving somewhat independently. And again, just to recap, it's actually probably not caused by quote-unquote epigenetic inheritance or even differential methylation in and of itself. We think that there's a broader regulatory mechanism, probably caused by differential inheritance of small RNAs, but we're testing that right now. Okay, so our next steps obviously are to let my computer finish pseudo-aligning the RNA-seq data. That would be cool. We also really want to do some population genetics of transgenerational plasticity. So we'd love to, when we get genes that we think are involved, we'd like to look for signatures of selection in these genes. I just want to give a quick plug for another project I'm working on, using machine learning to integrate genomic and epigenomic signals to look for this signature of selection. And obviously, we want a better understanding of fitness in the field. So we'd like to take these lines. I didn't mention this, but all of the genotypes that we used are parents of QTL mapping lines. So we'd like to put QTL mapping lines in the field and use that also to figure out what the fitness consequences of being able to express these different components of transgenerational plasticity is in nature. Okay, thank you so much for listening to this very dense overview. There's a lot of people to thank. Of course, my lab has been awesome and very supportive. Kathleen, of course, has been amazing. Cynthia Rudin has helped with the stuff we're trying to talk about, and that's the bell. So I'm happy to take questions.
So just thinking about your cold and could you say a little bit more about your cold and warm
Yes.
Did you have any predictions about?
A little bit. So we took the warm conditions are cycling diurnal temperatures that we took from the average spring temperature of the northern German part of the Arabidopsis range, and then that was the cold temperatures, and then the warm temperatures are from the Italian end of the range. So what the end being is 24 day and 16 night for the warm, and then 18 for the warm and 10 for the night. So they're not actually, yes, they're not actually that much different, and they do overlap a little bit. So to see, we actually predicted that the effects. We were a little afraid that we might not detect effects, but we actually do see in a lot of cases pretty strong.
So they were spring temperatures.
Yeah, they were spring type conditions.
So it was, all right.
Yeah, we didn't change the temperature over the course of the experiment, which we thought was like a whole new layer. Yeah, that'd be great.
So in your introduction, you talked about the matching between periodicity of the fluctuations in the environment and the generation times.
Yeah.
Thinking about the grand parental effects in that cycle, do you think that those effects are being imposed during the juvenile stage or during the flowering stage, and then you try altering when you're imposing the hotness as opposed to seeing?
Yes, so I didn't talk about this, but we actually we have this same experiment, but instead of thermal regime across the whole life cycle, we just changed fertilization. And so that only occurs during flowering. And then obviously the Q is relieved and the seeds are developing in just common garden conditions. And in that situation, we also saw transgenerational plasticity. They basically, it's the same results here, but just a little bit weaker. So I know that doesn't totally speak to, it would be really great if we could have also imposed an early life condition, like a reproductive exposure, and then see if the reproductive tissue, if it all developed in the common garden, we still see those effects. But we haven't gotten to that. That's a really good question. That would be really cool.
What kind of samples are you going to analyze for the smaller arrays?
Sorry.
What kind of samples are you going to analyze for the smaller arrays?
So for the small, okay, so for this first pass on the experiment, we took just Colombia, which has a lot of transgenerational plasticity that's very contingent on environmental sequences. So we have 12 biological replicates from each of those environmental sequences, and each of those samples gets total gene expression, small RNA, and DNA methylation for the sequence. So yeah, we're hoping to see differences between environmental sequences in the small RNA profiles. We think very specifically the 21 to 24 nucleotide small RNAs are going to be the most useful because those are the ones that are participating in the RDDN pathway. But we don't have that data yet, so I can't speak to it. But that's our hypothesis. It's based on the DNA methylation data, which I should also say occurred mostly in transposable elements, which is why we think RDD is